I think now I should say good afternoon, everyone. First of all, I would like to thank Dr. Klubal for giving me the opportunity to discuss here our current understanding on the molecular and cellular mechanisms of allergic diseases. So today I would like to talk about two different types of allergic diseases and also two different mechanisms I will show. There is actually two different types of allergic diseases, we say, and the third one I will not mention here. And we call it monoallergy or allergic breakthrough. So it's specific with the high, it's characterized with the high specific IgE to the certain antigens. And usually we do not see organ involvement in this type of allergic disease. And in the most cases that there is also no typical organ involvement, and those patients can have beneficial effect from specific immunotherapy with high success rate. So the example we can give to those type of allergies is venom allergy, latex allergy, or peanut allergy. So insect venom allergy causes 200 deaths in Europe every year, and it affects 4% of the whole population. So it causes reactions, local reactions, till the anaphylactoid reactions, which requires emergency treatment. Allergic response or allergic diseases are very well-known diseases also in the history. First report is coming from Egyptian time, and the King Menes of Egypt actually died because of wasp allergy in the 2621 before Christ. And they just resemble this animal to the wasp, they say. And the most common allergic disease is we call true atopy, and the patients show usually polyallergy to the different type of allergic molecules, and the disease we can describe as atopic dermatitis, asthma, and allergic rhinitis. And the disease is characterized with high total serum IgE and also high specific IgE to many allergens, and commonly eosinophilia involved to these allergic diseases. So another interesting story from the past showing us the genetical background of the true atopy. It's a Roman emperor family. So Emperor Augustus suffered from bronchial asthma, seasonal rhinitis, and atopic eczema. And Emperor Claudius had type of rhinoconjunctivitis, and his son Britannicus had horse dander allergy, and with swollen eyes and running nose. They say that actually he couldn't find the stable, and he was driven to the cage of the lions, and he lost his life there. And another interesting story is coming from England. King Richard III had strawberry allergy, and he used this allergic response to actually arrange the murder of his enemy, Lord William Hastings. Before coming to the Senate, he ate strawberry and developed acute urticaria. And he told in the Senate that actually Lord Hastings, put a curse on him, and Lord Hastings, this case, of course, Lord Hastings' life. how allergic inflammation starts. So first, we have to know that the allergic inflammation has four cardinal features. The first one is the activation of the effector cells. How they could be activated, and the activators could be air allergens, food antigens, autoantigens, superantigens, and yet unidentified factors. When the effector cells are activated, they should go to the target organs, which is very well controlled by the chemokine network, which defines actually the type of the disease. Either it would be atopic dermatitis or allergic rhinitis or asthma. When the effector cells reach to the target organs, they should survive in the target organs as long as possible to cause the inflammation. So during this prolonged survival in the inflammation area, second step of the activation occurs again with the allergens or superantigens. So the last step is the showing the effector functions of the effector cells and the like inducing of hyper IgE production or eosinophilia or killing the organ cells. So the first step is activation, homing, survival and reactivation, and the effector functions of the effector cells. If you go to the allergy congresses, you will see that the balance is always towards to Th2, and the allergologists try to change the balance toward to Th1. But if you go to the autoimmune system-related congresses, and the balance is on this side, and they always try to change it to T helper 2. What are those cells? When they are activated with certain molecules, they differentiate to T helper 2, which are characterized with the secretion of IL-4, IL-5, IL-6, 9, 13, and IL-25. If they differentiate to T helper 1 and they secrete interferon gamma and TNF alpha beta. So this, we thought for a long time that the only two different types of T helper cells we have. I will not say recently, but 3 decades before, we start to talk about the suppressor or regulatory cells. That time actually it was a little bit forbidden to talk about them because suppression was not accepted in that time. And also there were another difficulties to show those cells, to proliferate those cells, because they also suppress their own activation. And anyway, nevertheless, we have T regulatory cells, and they are characterized by the secretion of IL-10 and transforming growth factor beta. But of course, those two, together with those two soluble molecules, they also have surface molecules which works on their suppressive functions. What are the role of those cells in the system? T helper 1 cells are mainly responsible from delayed type of hypersensitivity reactions. They activate macrophages and they help B cells to calm down. And the T helper 2 cells help mainly B cells, eosinophils as survival factors, and also they stimulate mast cells. And T regulatory cells suppress both type of T helper 1 and T helper 2 cells, inhibits monocyte macrophage activation, and also inhibits dendritic cell maturation. You will hear many times the word apoptosis from me. What is apoptosis? Apoptosis is driven from a Greek word and means that the falling of the dead leaves from trees during fall. And apoptosis is very important, very essential mechanisms in the homeostasis of the cells and maintenance of the multicellular organisms. You will see following slides where the apoptosis playing very important role. I will show in atopic dermatitis and asthma, and also you will see where the cells are going either activation-induced cell death, where they are very much activated, or they will go under apoptosis when they have lack of survival factors. So for the maintenance of the system, apoptosis, I told you, is very important. Or survival factors and apoptotic factors should be in balance. If there is any defect in survival or increased, or apoptosis is decreased, we see lymphocytosis or lymphadenopathy like in infectious mononucleosis. If there is a defect in survival or increased, apoptosis is increased, we see lymphopenia like in HIV infections. What is going on in atopic dermatitis? And for a long time we didn't have specific molecule for asthma or for the allergic rhinitis, but we were very lucky when we started to work with atopic dermatitis because we have a specific molecule for the cells which home to the skin. We call this molecule cutaneous lymphocyte associated antigen, and the cells are CLA positive. What type of cells are they? What is the features of those cells? And they are in vivo activated. When we purify those cells, when we put them in the culture without any further stimulation, they show proliferative response. And they secrete T helper 2-like cytokines, like IL-5, IL-13, and they induce IgE mainly by IL-13 and prolonged eosinophil survival by IL-5. When we look at atopic dermatitis patients, although they have those CLA positive cells which are activated, and also when we look at this atopic dermatitis patient's skin, we see massive mononuclear cell infiltration. But in the periphery, there is no lymphocytosis. They have always normal lymphocyte numbers. So we ask the question, what is the mechanism which controls lymphocyte number in atopic dermatitis? And of course, we looked at the apoptotic features of those cells. We purified them from atopic dermatitis patients, also the counterpart CLA negative cells, to compare the survival of those cells. And for comparison, we also purify the same subsets from healthy individuals, from psoriatic patients, intrinsic type of asthma, bee venom allergic individuals, contact dermatitis, and intrinsic type of atopic dermatitis patients. So when we look at the survival of those cells after 48 hours, indeed, CLA positive T cell survival was much lower than CLA negative cells in all cases. But when we look at CLA positive cells from atopic dermatitis, and the survival time was much lower than the CLA positive cells which we purified from healthy or psoriatic, intrinsic type of asthma, bee venom, contact dermatitis, and intrinsic type of atopic dermatitis. So CLA positive cells which are coming from atopic dermatitis are showing low survival time point, or they die much quicker than other cells, other CLA positive cells. So we said that this activation-induced cell death on CLA-positive cells in atopic dermatitis could be the mechanism which controls the number of those cells in this disease. Till now, I talk in the periphery of atopic dermatitis patients, but what is going on in the skin where we see the disease? And this is the biopsy specimen of the atopic dermatitis patient. Then the disease is chronic, ongoing inflammatory skin disease, and 10% of infants and 5% of adults are affected in Europe. And when we look at the histopathology of the disease, which is characterized with the mononuclear cell infiltration in dermis, and the mononuclear cell infiltrates contain 70% of T cells, 1 to 3% of eosinophils, 10 to 20% of dendritic cells. In the dermis, we don't see NK, natural killer cells, or B cells. And the ratio between CD4 and CD8 is similar to periphery. It is 2. It means that CD4 positive cells are 2 times more than CD8 positive cells. And in the epidermis, this is CD4 staining. You see some CD4 positive cells. I will tell you later why it's important to see those cells, because they should be actually in the dermis. It means that they migrate to the epidermis. And here we see Fas and Fas ligand, which are very important death molecules. And it's hematoxylin eosin staining. It's the biopsy sessions of the atopic dermatitis patients. It's fast staining, it's bigger magnification, fast ligand staining, and also with bigger magnification. When we look at mononuclear cell infiltrates, we see very nice fast expression from probably T cells. 70% of the cells are T cells. But also fast expression from keratinocytes is very visible. When we look at fast ligand expression, again, mononuclear cell infiltrate express very high fast ligand, but keratinocytes do not express fast ligand. So in the dermis, mononuclear cells express both death molecule, fast and fast ligand. In principle, they should kill themselves. And then now here we see hematoxylin eosin staining. At the same area, we stain with tunnel staining, which shows the apoptotic nuclei of the cells. And Hoek staining is another staining which is showing apoptosis of the cells to us for the complementing of the tunnel staining. And here we see keratinocytes in the epidermis and mononuclear cell infiltrates in the dermis. When we look at tunnel staining, although we do not see any apoptotic cells in the dermis in mononuclear cell infiltrates, we see very nice keratinocyte apoptosis in the epidermis with the red stained nuclei of apoptotic keratinocytes. When we look at Hoek staining, we see similar results, no apoptosis in the mononuclear cell infiltrates, but we see keratinocyte apoptosis. So it means that mononuclear cells in atopic dermatitis are resistant. In a way, they're resistant to apoptosis, although they have all apoptotic molecules on their surface. In atopic dermatitis, we see very important changes on the cytokine profile when we look at the early time point and the chronic phase of the disease. Early time point, especially atopy patch test after 72 hours, which shows very high IL-4 secretion, goes almost to 0 after 3 to 6 days. And very interestingly, interferon gamma was very limited in the beginning, but on the late phase, interferon gamma goes up, and IL-4, IL-5, and IL-13 do not show so big difference. If we see some changes, that could be some little decrease. I will tell you why it's important to see interferon gamma in the system, because till now, when we talk about allergic diseases, we always told that they are Th2 related diseases. But actually in the system, during the tissue injury, interferon gamma is giving much more harm than IL-4 or IL-13. And those experiments were set up in our laboratory by Axel Trautmann, and he used primary human keratinocyte cells, and this is the monolayer of keratinocytes, and he treated those cells with either resting T cells or activated T cells. He activated the T cells with antigens or polyclonal stimulation like anti-CD2, 328 monoclonal antibody mixture. What we have seen here, when we add resting T cells to the keratinocyte cultures, we don't see anything. But when we add activated T cells to the cultures, keratinocytes are getting round and detach from the plate. When we stain those cells with the apoptotic staining like Hoech's and resting T cell related conditions, we don't see any apoptosis, but when we add activated T cells, we see very nice bright condensed nuclei of apoptotic keratinocytes. He has done the same experiment with the human skin equivalents, and here you see hematoxylane staining, and here you see tunnel staining. And again, he manipulated the skin equivalents with resting T cells or with activated T cells. When he add activated T cells to the cultures, we see very nice red stained apoptotic nuclei of keratinocytes. And working with the neutralizing experiments showed us actually there is 3 different types of molecules which play very important role in the apoptosis of keratinocytes. They were interferon gamma, Fas ligand, and TNF-alpha. And it's very important to know those molecules to develop better treatment for allergic diseases. I would like to conclude the first part of my talk with this figure. Effector cells, I told you in the beginning that they transmigrate to the skin or in asthma to the lung. And the inflammation, allergic inflammation, in the beginning, it's antigen specific. We all know that. But when the inflammation is becoming chronic, it's not antigen specific anymore. It is antigen non-specific because when they wanted to clone all T cells from skin, and only 1% of the cloned T cells showed specificity to the initial antigen. So the rest were not specific to the initial antigen. The major players are T cells in the skin and T cells and eosinophils in the lung. And the effector molecules were interferon gamma, TNF-alpha, Fas ligand. So now I will talk about interferon gamma, why it is so important. Because if you measure the amount of interferon gamma in allergic diseases, you might not find so high amounts of interferon gamma, but you might find very high amounts of IL-4, IL-13, or IL-5. But interferon gamma is evolutionarily very well developed cytokine. To kill keratinocyte with interferon gamma needs only 1 nanogram per ml. But to induce IgE by IL-4 or IL-13, you need 50 nanograms of those cytokines. So little amount of interferon gamma can cause the tissue injury. So it's important, still we should think that changing the balance towards 2 T helper 1 will not be helpful for the treatment of those allergic diseases. And interferon gamma is responsible from the apoptosis of the keratinocyte, which shows the spongiform morphology and leads the eczema formation in atopic dermatitis. And again, interferon gamma kills bronchial epithelial cells and responsible from bronchial epithelial cell shedding in asthma. In the very severe cases, we see also smooth muscle cell apoptosis. Of course, counter-regulation comes from epidermis cells from keratinocyte to the T cells or eosinophils to migrate to the epidermis and to cause more problem in the disease. So I will move a little bit to the totally different area to your mind, also my mind, but you will see at the end that this mechanism is also very important to or into the pathogenesis of allergic diseases.
So until now, your conclusion is that cell death might be important for allergen tolerance?
Yeah. Yeah, not allergen tolerance, but dysregulated apoptosis in the allergic diseases is very key mechanism how the disease chronicity goes on.
The data that you showed on atopic dermatitis, were there any data published on lung tissue as well, the same?
Yes,
yes.
Yes. And this part is already published. If we would like to describe some diseases in a very simple way, we say allergy is intolerance to allergens. Autoimmunity is also very simple, intolerance to autoimmune antigens. And transplantation rejection, we have problem to tolerate transplanted organ. But on the other way, is tolerance good all the time? And we don't think so, because during the chronic infections, we have problem to neutralize the infectious agents from the body. And we can give an example here, like cystic fibrosis patients, they tolerate Pseudomonas antigen instead of fighting with those. And in some type of cancers, we know that there is inappropriate immune response to tumor antigens. Even around the breast cancer cells, they showed high IL-10 secretion that tumor cells can protect themselves from CD8 positive cells related clearance. So our first studies in this area, we wanted to know the peripheral blood mononuclear cell response to the major allergen phospholipase A2 in ultra-rush bee venom specific immunotherapy. When we receive peripheral blood from those patients, it was before immunotherapy, 7 days after immunotherapy, and nearly 1 month after immunotherapy. What was the finding, very important findings in this study? The proliferative response to phospholipase A2 decreased during the immunotherapy. And Th2 cytokines like IL-4, IL-5, IL-13 also decreased. And T helper 1 cytokine, interferon gamma, also decreased. In the same culture, only 1 cytokine, IL-10, showed very significant increase. And we asked the question, if those effects are really related with IL-10, production of IL-10, so we neutralize IL-10 in the system. Indeed, T cell proliferation comes back together with cytokine secretion. We asked another question, what was the source of IL-10? And we gauge CD4 positive, CD25 positive cells by flow cytometry, but we didn't think that they were regulatory cells that time, and we gauged them because they were effector cells. And IL-10 content of those cells was 9% in the beginning of immunotherapy, and it goes up to 30% after 1 week of immunotherapy, and 50% after 1 month of immunotherapy. This is another immunotherapy study which was applied to the DERP1 allergic children. And we collect blood samples day 0, 7, 28, and 70. And we looked at the cytokine profile of those samples, and we see a very nice decrease on IL-5, IL-13, and interferon gamma. Only IL-10 and TGF-beta showed very significant increase in the same cultures. So we got the idea that the, as a model immunotherapy, we could show some suppressor cytokines or suppressor molecules, and this gave us some idea how could be the balance, or if the balance is decisive to give healthy immune response or allergic immune response to the allergens. And this was our hypothesis. Before specific immunotherapy, and allergic individuals, probably cell proportions should be like that. They have huge number of T helper 2 cells, but they also have T helper 1 cells. Probably they have small portion of T regulatory cells. And after successful specific immunotherapy or healthy individuals, they have different proportions of those cells. They also have T helper 2 cells, but with a little number, and they have T helper 1 cells, but they have probably higher numbers of T regulatory cells. And our aim was to prove this hypothesis. And when we start to study bee venom allergy 12 years ago, it gives us actually a chance to work with also healthy immune response to high dose antigen exposure on beekeepers. Our beekeepers expose with high number of or high amounts of antigens, indeed, because they have more than 20 bee stings in 1 week. Beekeepers in Davos start to collect honey in the beginning of May. So during all those years, we took 100 times of blood samples from them in a different time point of the years. What we realize, when they start to collect honey, when we look at the phospholipase A2 specific T cell proliferative response, we were not able to stimulate the cells. And cells did not respond to phospholipase A2 till the end of September, where the season was ending, and they could reach the initial proliferative response that time if they are not stung. So this was a very unique model for us, and the findings on beekeepers we didn't publish yet, and on the cytokine secreting cells. So recently developed technique enabled us to purify antigen-specific cells according to their cytokine profile. But to look at the frequency, you don't need to purify the cells. You can use another technique, which we call ELISpot. You can stimulate the cells with the certain antigens, control antigens, and you can look at the cell frequency. What we have done in beekeepers, we look at the phospholipase A2 specific IL-4 secreting cells, IL-10 secreting cells, and interferon gamma secreting cell frequency before they are stung. IL-4 secreting cells represent T helper 2 cells, interferon gamma secreting cells represent T helper 1 cells, and IL-10 secreting cells represent TR1 cells, which T regulatory cells. And only 7 days after they are stung, they receive multiple bee stings. IL-4 secreting cell frequency was already low, but it goes down. And interferon gamma secreting cell frequency was higher than IL-4 secreting cell frequency, but it also goes down. And only IL-10 secreting cell frequency goes up when they receive high amounts of antigen. So they are healthy, they don't show any allergic immune response, but they have the cell type which is responsible from allergic immune response. They are not deleted. And after having this evidence that our hypothesis could be true, and we looked at allergic individuals and healthy individuals' antigen-specific cytokine secreting cell frequency. We use antigens like DERP1 from house dust mite, BETV1 from birch, PIRC5 from pear, CORE1 from hazelnut. So when we look at allergic individuals first, IL-4 secreting cell frequency was much higher than interferon gamma and IL-10 secreting cell frequency. When we look at healthy individuals, they have lower proportion or low proportion of IL-4 secreting cells and a little bit higher interferon gamma secreting cells, but with much higher frequency of IL-10 secreting cells.
So they have naturally more irregular cells?
Yes. Yeah. I mean, all of us who doesn't show any allergic immune response to the allergens. And this is the mechanism actually how they protect themselves. And this figure shows many things to us. First of all, for allergic individual to have IL-10 secreting cells explain us that the specific immunotherapy could be successful if the patients could be chosen properly. And in healthy individuals, we have bad news because they have the capacity to develop allergic immune response to every kind of antigens one day in their life. And this figure explains us how we have seen monoallergies in the individuals who were healthy before. Because we have IL-4 secreting cells which are specific to the antigens. So this is the mechanism. If you have any questions, we can really talk about this.
Can naturally return back the allergy disappears?
I think yes. What happened actually in your case? Because you were allergic to apple and the hazelnut, and they were both disappeared after a while. Probably you didn't avoid to eat those antigens, did you? The food to eat, yes. This is the way actually how we restore the peripheral tolerance.
But what happens if we treat the patient immediately with immunotherapy? That you have an allergic patient who was before healthy, we give the patient immunotherapy. What happens? Do we treat the patient? Is there a danger that the patient switches to more allergic person?
In monoallergies, actually, immunotherapy is much more successful than polyallergic individuals. So they suggest, for example, bee venom allergy or bee venom specific immunotherapy is successful 95%. But the problem is that the immunotherapy takes really long time, and you don't know when you need to cut it. And why the frequency of the cells are important for us. And we have to know that we are dealing with the ongoing chronic disease as allergic diseases. So when our patients comes to the polyclinic, we never see the sensitization phase of the disease. It's already gone. And we see ongoing memory response. And the memory response is important. It is the feature of the T cell and B cell specificity that when they receive high dose of antigen or the antigen, they should respond rapidly, and both T cell and B cells. So when the antigen is presented by the professional antigen presenting cells, like dendritic cells, it's very important what could be the first cells. Is it going to be a T helper 1 cells which will lead more tissue injury and the chronic phase of the disease? Or is it going to be a T helper 2 cells which will cause more allergic inflammation and also hyper IgE production? Or is it going to be a T regulatory cells which will silence down the many different aspects of the immune system? And T regulatory cells, when we purify them according to the IL-10 production, we looked at what type of suppressor molecule they use. Are they only using IL-10 and TGF-beta, or there is another suppressor molecules? The IL-10 content was already high in those cells, but they also have IL-10 receptor on their surface. It means that they can use their own IL-10 and they can limit their suppressor functions as well. And CTLA-4 is cytotoxic T lymphocyte antigen 4. It is a suppressor molecule they express very highly on their surface. CD25, they were positive because we actually stimulate them with the antigen. And program that molecule 1 is another suppressor molecule they have on their surface. And TGF-beta receptor 1 and receptor 2, we stain after cytospin of the cells, they were both positive. So as IL-10, we know that suppresses Ig and induces IgG4. Our recent paper is nearly ready to publish. When we purify different type of T regulatory cells on cell base, we showed that they suppress Ig and induces IgG4. And TGF-beta also suppresses Ig and induces IgA. So when we show that the IL-10 is in allergic individuals, and then the peripheral tolerance mechanisms also important for the allergic diseases, and there could be a break in this mechanism. And we actually, in the beginning, we were thinking that if we are able to show lack of IL-10 or IL-10 receptor in atopic dermatitis patients, and we would be able to show the real mechanism why they have a disease. But of course, nothing was so easy, and we were not able to show lack of IL-10. Actually, in atopic dermatitis patients, they secrete much more IL-10 than the normal individuals, and they have also TGF-beta. And we said, okay, they secrete the cytokines, but maybe there is lack of receptor, they cannot use the cytokines. But we were wrong, because they also have the receptor. They can use IL-10, they can also use TGF-beta in the system, but still there is allergic inflammation in the skin. So it wasn't so simple, luckily. And what we wanted to show that if there is any problem for the transmigrating of those cells to the skin. And you may heard about CD4 positive, CD25 positive naturally occurring FOXP3 expressing cells. So we thought that maybe there is something wrong with those cells, and although we have shown that in the periphery they have FOXP3 positive, CD4 positive, CD25 positive cells, but what is going on in the skin? And when we stain the atopic dermatitis skin, atopy patch test and psoriatic skin, they were all CD25 positive. And when we look at FOXP3 staining in those samples, they were lack of FOXP3. There were no FOXP3 positivity. And to be sure that our staining is working, we took the positive controls like contact dermatitis and tonsil, and we see very nice FOXP3 staining here. And to be sure that our molecule is specifically staining, and we suppress the FOXP3 staining with the specific peptide. And indeed, the staining was specific for the FOXP3. And I would like to conclude my talk with this figure. T regulatory cells, they are allergen specific, and they use multiple suppressor factors. When they are activated with the help of dendritic cells, and they upregulate IL-10, TGF-beta, both receptors, CTLA-4, PD-1, and our recent finding was histamine receptor 2, and suppress directly effector cells because of production of IL-10 and TGF-beta, and together with the direct suppression show indirect suppression too, because T regulatory cells can suppress T helper 2 cells, which provide cytokines and survival factors for the effector cells. And T regulatory cells help dendritic cells, if they are immature phase, keep dendritic cells in the immature phase, and if they are mature phase, tells them that they make less IL-12 but more IL-10. And T regulatory cells with the IL-10 and TGF-beta help B cells to induce more IgG4 and IgA. We call them non-inflammatory antibody isotypes, and tell B cells that they produce less IgE. I would like to thank to my co-workers, of course. Without their help, those studies would not be done. And I would like to invite all of you to our World Immune Regulation meeting in Davos in April. Thank you for your attention.
Thank you very much. Máte otázky někdo? Už bude jídlo. I have one question. It's connected with next part, which is going to be after lunch. If I have antigen presenting cell, and T cells, as you described it, Th1, Th2, and T regulatory cells, what is the optimal pathway so that it results in no allergy? Biochemistry of antigen, presentation antigen presenting cell, and I connect it with the next part because we talk about immunotherapy. So how to give the patient the antigen, in what kind of medium, so that it results in upregulation of T regulatory cells, and it's not just that by chance it happens that we expand T regulatory cell. Is it the recombinant form of antigen or mixture of antigen with some kind of glyco structures?
So let me think to respond this. The antigen what we are using in our cultures are recombinant. When we suggest that to use recombinant antigens in the cultures, of course, it's not always available. And our friends tells us that, but in the immune system, we also have LPS or other innate immune system triggering molecules at the same time point when we expose with the antigens. That is right, but antigen presenting cells present everything, because actually antigen presenting cells cannot decide if we have a molecule which causes danger or it's harmless. And the antigen presenting cells should present everything to the T cells. And T cell will decide if it is danger, containing danger signal, or it's harmless. So in the experimental level, if we have LPS contaminated material, of course the result will not be pure, and even we will have difficulties to interpretate the results, which is, or what part is coming from antigen itself or LPS or other contaminants. So we suggest that we should use recombinant antigens as much as possible, and whatever we use in our laboratory, we first measure all LPS content with the LAL technique. And what else I would like to say? Also in the specific immunotherapy, I think allergopharma is driving recombinant allergen immunotherapy for grass pollen, and they try 5 different grass pollen allergens, which was recombinant, and combine 5 different major allergens in one vaccine, and the study is still going on. I guess also the companies are aware about the contaminants or extra problems, because we all know that with vaccine we can sensitize the patient's other molecules too, if it is not pure.
Thank you very much. Máš někdo otázky nějaký? Tak děkuju moc krát.